Wonderful. Okay. So this sort of is a I guess you could say this is a sort of intentionally antagonistic title. Of course, insect cuticle should be studied not just by entomologists but also by engineers. But it's gotten a lot of attention recently from engineers, especially, and not so much on the biological side. So recently, I was in Japan for the International Congress of Entomology, and there was a section on insect cuticle, and all of the talks fell into either two categories. Talks where somebody had done some kind of mechanical experiment on a piece of cuticle or a particular or a special cuticular structure in an arthropod, and then they presented the results of these mechanical tests, or they were studying the development of the cuticle and were presenting like genetics, basically developmental pathways that they had worked on. And so what's really missing, in my view, and this is what I'm going to expand upon a little bit here with this presentation, is actually a deeper understanding of what makes a cuticle, what the different parts of it are. Because if you look between different insects and different arthropods, it's not always immediately apparent which parts of the cuticle in cross section are homologous, like which parts belong to the exocuticle or endocuticle, what different types of layers are there, and you'll see what I mean when I show you some pictures. And that sort of good old-fashioned morphology work is really missing in this character system for most arthropods. It's actually quite shocking as a person who studies morphology. So we need more scholarship in that area, you could say. But first, we'll kind of get into why people are interested in cuticle in the first place. Insect cuticle is really special, or arthropod cuticle in general is rather special because it's a type of composite called a fibrous laminate composite. These are things that only in recent years have been adapted for use by humans. So if you've ever worked with like fiberglass or carbon fiber composites, I don't know if any of you ride like mountain bikes and they're built out of carbon fiber and they have to be optimized a certain way to distribute loads from your body. Or if you make a boat, right, out of a shell out of fiberglass, you have to arrange the fiber layers in different orientations so that they're not all facing the same direction, so that if you hit something, the load is distributed more evenly across the surface, right? The thing is, insects do the same thing. So the picture in the background here is actually from a weevil. Beetle exoskeletons are fairly special, even for insects. And this has gotten a lot of attention from engineers recently because it's what's called a hierarchical material. So in addition to being a laminate composite, so we'll define some terms here. A laminate composite is a type of material that is made up of layers. So each layer has either a different orientation or a different type of structure or a different type of material. So anytime you just layer things together, it's a laminate. A fibrous laminate composite is one where all of the laminae, or the layers, are made up of fibers that are embedded into some kind of matrix. So in man-made systems, you would see glass fibers, for instance.
Enhanced program.
You'd see glass fibers embedded in like epoxy, for instance. Same thing with like carbon fiber composites. You would have carbon fiber sheets that are embedded in some kind of an epoxy or other resin, like a high temperature resin or something. In biological systems, such as arthropod cuticle, the fiber in this case is chitin, right? So alpha chitin in most cases, and it's embedded in a protein matrix. But it's a little more complicated than that. In insects, in arthropods in general, the cuticle is called a hierarchical material because it has multiple levels of structure depending on what length scale you examine the material at. So at the very smallest level, at the level of molecules, the fiber is alpha chitin, which is in this diagram here is, I don't know if you guys can see my mouse, but over on the left-hand side on the bottom at the 10 angstrom level, You can see chitin chains. These are arranged linearly into crystals. Those crystals are bound together with protein. And then those individual crystalline fibers are meshed together into larger bundles. And then those larger bundles are arranged into layers. And that's what actually makes the different layers inside of a cuticle. So there's multiple levels of structure. And the reason for this, as it turns out, and if you can see the structure, so we're going to be spending a lot of time looking at cross sections. So just so you know, if you take a piece of exoskeleton, so imagine you have like a lobster shell, you break a piece of the shell off and you cut it in cross section, right? So if this is my lobster shell, I cut it this way, and I'm now looking at it in a side view, if you will, in cross section. What you can see is that there are distinct zones inside of the cuticle. So there is, generally speaking, what's called an endocuticle and an exocuticle. Those are very coarse terms. They don't mean anything really other than, well, we have an innermost layer and an outermost layer. That's it. There's no real homology there. Nobody knows if the exocuticle of a lobster corresponds to the exocuticle of a beetle, if they're derived evolutionarily from the same structure. We don't know that yet. Nobody's tested that. So at this point, those are very coarse terms just to describe position. We have a layer that's towards the outside. We have a layer that's toward the inside. Sometimes there's more than one zone. Sometimes there are several. But we'll get to that. Part of the reason that these materials get a lot of attention is because they can be incredibly strong. So this beetle here, for instance, some of you may recognize this is Zopharis, the ironclad beetle. There was a lot of attention recently in the media. There was a person from, was it UC Santa Barbara? No, somewhere. It was somewhere in Southern California. I forget which institute. Got a bunch of money from DOD to study this because this beetle can be run over by a moving vehicle and not be damaged, which is incredible. These are very, very strong insects. If you've ever tried to pin one of these, a very common phenomenon is that the pin will just bend as you press the pin into the elytra. And so they were studying this, to see just how the cuticle is able to resist being, deformed when it's run over by a massive amount of weight or force in this case. And when they did this, they focused mostly on the shape of the elytra, the interlocking mechanism, but the underlying arrangement of the different layers of the cuticle was completely ignored, wasn't accounted for, which to me was a bit disappointing. Now, as far as the layering types that are involved, I think most people are familiar with this organism here. This is a mantis shrimp. It's thought that having layers inside of the cuticle and this hierarchical structure helps keep the cuticle from breaking. It just prevents the fibers from splitting or cracking, right? Imagine if I had a sugar cube and I took the sugar, right, which can be a linear molecule if you arrange it in a certain way. If I just took a sugar cube and hit it with a hammer, right, it's going to break apart into a million pieces. But if I link all of those glucose molecules or if I link a bunch of carbohydrates into something like, say, cellulose, and I embed it with a little bit of protein, that's like, you could then imagine a piece of wood of the same size. If you bang on that with a hammer, nothing's going to happen, right? Because it's much stronger, because you have that chain, you have those chain structures, and then it's also arranged hierarchically in the same way that an insect exoskeleton is. Makes it a lot stronger. So I think, let me see, I'm just going to play this very briefly. Oh, Lord. Okay. There is one thing I want to show you from this video, if I can find it. Here. So it uses these two clubs. They're called dactyl clubs. They're very well known for being high speed, for being high speed impact hammers, basically. And they use them to crack open shells of things like snails, crabs, other small crustaceans. So these are predatory animals, and they have these dactyl clubs, and they absolutely just nail things with them at very high speed. And the question, of course, from an engineering perspective is how are these structures able to resist cracking when, you have something like this club and it's impacting a calcium carbonate composite, in this case, a snail shell, and it's able to break the snail shell without itself being broken. And the way that they're able to move on. The way that this works is that all of the layers inside of the cuticle are arranged at a very small angle relative to the adjacent layers. And so you get something called a helicoidal ply layup, which in insect exoskeletons is referred to as the Boulogne structure after the person who discovered it first. This structure is, well, if you can see my mouse in subfigure C here, you can see how this kind of works. You have a single layer, a single sheet of chitin fibers, and then the layer above that is very slightly offset, and it makes this kind of helicoidal arrangement. By having that arrangement, anytime a crack initiates in the material, the crack has to reorient at every successive layer in order to progress through the material, in order to propagate. So it becomes very difficult to actually crack this material. This arrangement imparts a high degree of what's called toughness, so the ability to resist fracture. And this has been studied ad nauseum by a lot of people, the Boulogne structure and crack propagation. And so it's thought that this sort of architecture prevents cracks from forming, and it reduces the brittleness of the chitin. So again, chitin is a sugar molecule. By itself, it's about as brittle as like the sugar in a sugar cube, right? It's very stiff. It has very high stiffness, but it's very brittle, like glass. So having it embedded in a resin and arranged in this way makes it tough. So you can use the high stiffness, but without having to worry about it breaking or shattering at the slightest touch. So as a PhD student, I studied this in beetles because beetles are a little bit of a special case. They have a Boulogne structure in part of their cuticle, but in another part of their cuticle, they don't have this at all. They do something completely different and weird. So this organism right here is female Curculio-sayi, the lesser chestnut weevil. So this one feeds on chinquapins and oh god, what are they called? Like horse chestnuts, and also like regular American chestnuts when those were around. As you can see, it has a very exaggerated structure attached to its face. This is called the rostrum, and at the very tip of the rostrum, so you can kind of imagine this as a tube with a sphere attached to the end of it. The whole thing is hollow. So the head with the eyes is over here. There are muscles inside of this head that actuate the mouth parts, which are located up here at the tip. Those little mouth parts are what actually cuts stuff. And then the rest of the rostrum is basically just there to keep the mouth parts at the tip more or less like a laparoscopic device. These organisms use the rostrum with these mouth parts all the way at the tip to make or to excise oviposition chambers for their eggs. So what they do is they'll sit on an acorn or some other type of nut, depending on the species. There are, I think, 35 species of curculio in America north of Mexico. And if you include Mexico, I think that jumps up to like 80 species or something. So they're quite diverse. They get on a lot of different kinds of nuts, but all of them use the rostrum to excavate oviposition sites. So they will drill a hole into the acorn. And I guess my contribution to science was discovering that the rostrum actually bends a lot while they're doing this. In this case, the rostrum will actually bend by up to 90 degrees and be completely straightened while they're digging out these holes. And what they'll do is they'll bury the rostrum in all the way up to the eyes, and they'll kind of wiggle their head around and kind of make this drilling motion. All of the chewing is happening, of course, here at the tip, but they're doing that just to kind of straighten out they'll make a completely arrow straight hole into the acorn, and then they'll turn around and they have a telescoping ovipositor. They'll lay eggs into the hole. So yeah, here's a close-up of a different species. As you can see, the whole structure is completely continuous, right? There's no joints. It's just a solid piece of exoskeleton. It's completely hollow all throughout. So this whole thing is hollow. Um, at the tip, you have a couple of mouth parts. This is all completely hollow, and this head is also completely hollow, just with some muscles in there and a couple of internal support structures to kind of hold the muscles and mouth part. Excuse me, the esophagus in place. Now, what's special about this is that the rostrum, as I said, can bend quite a bit, and I think this video works. There we go. I did a mechanical test on this to see, how robust is this? This is a head that was in a museum collection, probably about 40 years old. No, 40 years old? Was it 20 years old? This one was 20 years old. Completely dry. I took it off. I greased it up so that it would, I soaked it overnight, greased it up so that it wouldn't evaporate out. And then I hooked it up to a pneumatic a pneumatic testing device and wanted to see how many cycles until the thing would break. This went on for 3 weeks before the material science lab called me and said, hey, we'd like to turn this off because the bills are really running up for the electricity on this pneumatic device. So I think this went through a couple 100,000 cycles. Went to just about 400,000 cycles before we said, okay, enough is enough. It's not going to break. And what I'd like to point out here in this particular figure is that the structure never actually breaks. It does straighten out quite a bit, but it never actually breaks. We got to around 400,000 cycles without it fracturing. And afterwards, I did a mechanical test. I soaked it again in water to let it relax, because these are viscoelastic materials. They stretch over time. So I let it relax in water, and then the next day did a test on it to see if it was damaged, if it would snap the way that other rostra would, normally break. And it was exactly the same as all the rest of its conspecifics. So there was no microscopic damage that was visible. It behaved exactly the same mechanically after it was allowed to relax. These structures are really incredible for their robustness. And the reason for
Andrew,
this,
could I ask a quick question while you're on this?
yeah,
The device you had that was showing in the video, what exactly was the red portion there that was bending back and forth?
um,
The red.
oh,
Yeah,
this orange thing here,
yeah.
that's a piece of ripstop nylon. It has extremely high tensile strength. I wanted to make sure that what I was using was flexible and, so it was very flimsy. It wouldn't force the snout to bend. The snout is actually quite stiff. So I wanted to use something that was flimsy but had very high tensile strength so that whenever the rostrum would return to its original position, it was doing so by its own elasticity, not because something was pushing it back into position, if that makes sense. So yeah, that's what this is. Yeah, just ripstop nylon and it's glued in there.
So
Um,
it's pulling and then releasing. Is that what's going
yeah,
on?
exactly. And it returns to that shape that it has right now, that bent shape, just under its own elasticity. Yep.
Okay, thank you.
And the reason that this kind of works, part of the reason that this works, so this is where the microscopy comes into play. I cleared a couple of these heads and then split them with a razor blade. And splitting one of these things with a razor blade is not easy considering it's only 250 microns in diameter. But I did it, and I did some confocal microscopy on this to see if there were any differences in the composition of the cuticle in the tip versus in the back of the head. And what I found out was that in the apical portion of the rostrum, so that's on the left-hand side here, there is a much thicker endocuticle than in the right-hand side. So there are some differences in, and there's also differences in the way the fibers are arranged and things like that. Essentially, the cuticle, what I discovered was that the cuticle at the tip of the rostrum, the part that's actually bending, is optimized for bending. It is more flexible and has higher toughness than the stuff at the base of the rostrum and in the head capsule. That's optimized for rigidity. So it's very stiff, but also quite brittle. And the way that this optimization happens, if you look at the microstructure of these in cross section, so here's just a cross section through the snout. So the snout is, of course, cylindrical. So when you cut through it, you see a circle. Almost all of the thickness of the cuticle is endocuticle. Now, in beetles, endocuticle is a bit of a special thing. It's comprised of very large bundles of chitin, and they are not arranged in that helicoidal ply. So you just have discrete layers, very thick, with all the fibers facing in the same direction. And the endocuticle in this case also has something called resilin in it, which makes it a bit tougher and a bit less rigid. So there's a bit of a trade-off there. So the endocuticle in general is tougher but less rigid than the exocuticle. The exocuticle has that helicoidal arrangement, and it's very stiff, but by far more brittle. So in the area that's optimized for bending, you have a very thick endocuticle, a very tough material that is also quite flexible. And the stiff layer is reduced to a very thin region around the outside. So if you could kind of imagine, if I have, if you can take a very stiff material, and if you make it into a very thin layer, it will bend without breaking, right? That's what's more or less happening at the apex of the rostrum. On the right-hand side, the stuff that is optimized for rigidity, the endocuticle is maybe about 50% of the through thickness, and the exocuticle, which is much stiffer, occupies a correspondingly larger percentage of the thickness. So it's just generally a lot stiffer because of that. In addition to this, if you look at the endocuticle, there are effects from the direction that the fibers face. So these large bundles of chitin it's called anisotropic. They are much stiffer along the direction of the fiber, right, than in the off-axis direction. So kind of back up here a little bit. If you have a piece of wood, so imagine I give you a 2x4. If the grain of the wood is running lengthwise along that 2x4, it's going to be very stiff in that direction. And if you try to cut against the grain versus with the grain, you're going to get very different. Results, right? It's the same thing with insect cuticle. If you are pulling against those fibers, right? So if I had a bundle of these fibers and I was trying to pull and they're all aligned in the same direction, it's very, very stiff. So you won't get much elasticity in that direction, right? But if you do it in the opposite, if you do it across the fibers, then it's much less stiff. So having these super stiff fibers, what the what the weevil does is it arranges them in a 45-degree ply, where the longitudinal axis is offset to that. So in layman's terms, the fibers are turned away from the direction of bending so that they maximize the flexibility of the structure. And the 45-degree angle is as flexible as you can get with the type of layup that they have. At the base of the head, these fibers are arranged such that they face in every single direction. They're all over the place. So it doesn't matter what way you pull on it, it's uniformly stiff in all directions. So it exhibits what's called transverse isotropy. So combining these two characteristics means that the apical region is optimized for bending, the basal region is optimized for rigidity. And here's the thing. This is not a special beetle. It's not a special case. All insects display some degree of optimization in the microstructure of their cuticle. You can break open any set of structures on an insect and you'll see they are not the same from body part to body part. And it is completely unstudied. No one knows what's being done, what rules are being followed, or how this evolved. Completely a black box. I got a PhD for doing this. If you're a grad student watching this presentation, you could do the same thing. It's pretty cool. These are great things to see. And the thing is, when I did this, everybody told me, I was a biologist. I have a biology background, and here I am doing like composite mechanics and math modeling and finite element analysis. Everybody told me, yeah, these are engineering problems. You need to have materials scientist and an engineer on your committee, which I did. They were very helpful. But fundamentally, studying insect cuticle is not inherently an engineering study. These are biological systems, biomechanical systems, and the bio part is very easy to overlook. I cannot tell you the number of times I've seen an engineer come forth to study something in a biological system and draw some very strange conclusions because they ignored the biology aspect of the problem. So these sorts of systems have been studied only a couple of times, as I kind of alluded to before. This is a great example of a biologist who noticed something was amiss in an insect that he was studying. This is a guy named Loader. This is in 1992. Heteropteryx dilatata is a gigantic stick insect. They're like the size of a banana. They're awesome. They have very thick cuticle, very easy to study. This guy was studying cuticle development, but what he noticed when he started taking the insects apart is that the cuticle had a different type of ply structure, or a layer arrangement, if you will, in every single body part that he examined. And he made note of this and said, it looks like these could be mechanically optimized. That wasn't what he was going for in that paper. He was looking at development. But he made the observation and nobody picked up on it. Oh, there's the guy. Yeah. They're pretty cute. But yeah, they're huge. Same thing was observed in spiders. Somebody took the time to examine the number and arrangement of layers in the cuticle of different parts of spiders and in different species. Totally different. Nobody said anything. Crickets. So as a postdoc, having seen this, I kind of wondered, what happens if we compare a little bit between species? And it becomes very difficult to make meaningful comparisons if you can't say what the different parts of the cuticle actually are. So I had to back up a little bit and review the literature for this field and see, what terms exist and how the different parts of the cuticle are classified, because it varies depending on what taxon you're looking at. In beetles, this is pretty easy. This is a pretty easy thing to study because there's a discrete exocuticle and there is a discrete endocuticle, and they're very easy to tell apart. One of them is helicoidal. The other one has these massive, chunky fibers in it. In things like Polyneoptera, it's not always so clear-cut, as we'll see. So traditionally, the way that the different parts of the cuticle are delineated is through histology, through staining. The method that gets used nowadays when people do this is based on a protocol from Leo Schatz. It's in 1952. This is a modification of a Roberts 1950 protocol where he added something for what's called mesocuticle. So the basic protocol is you take a piece of cuticle, preserve it in Bowen's fluid, then mount a cross section of it on a slide, stain it with Mallory trichrome, wash it, and then if a part of the cuticle stains amber or doesn't have any stain whatsoever, that's the exocuticle. If it stains red, that's the mesocuticle, and if it stains blue, that's the endocuticle. And it's important to note, Leo Schatz said in his paper, he said it doesn't matter where the stain is, outside versus inside, doesn't matter. Wherever you see the color, that's what it is from a homology standpoint. That was his homology hypothesis. That doesn't always work. And I will show you the staining. The staining can be all over the place. I'll show you some pictures in just a bit. Because the staining is kind of inconsistent depending on what taxon you're looking at, and we don't really know what's being stained also. That's a bit of a problem. What's generally accepted is that the outermost layer that is helicoidal, that's the exocuticle. The inner set of thick layers, that's the endocuticle, right? That's generally accepted because if you're doing something like TEM or SEM, right? If you're looking through an electron microscope, there's no way to colorize it. It's black and white. So you have to look at structure rather than histology. So here's an example of these same three taxa. So I have a mole cricket, a stick insect, and a praying mantis. Yeah, these three species were my test species. And we have cross sections from those same three species looking through the TEM. And what you can see is that there are a large number of layers. And so my question to you guys out in Zoom land is, how would you try to make comparisons between these three sets of cuticle? Like, what parts are the same? That's a tricky question to answer. And what I kind of what I ended up going with here, I first tried to look at it with staining, and the staining is nonsense. The staining regimen that most people use is actually done in locusts. So Grillotalpa, being an orthopteran, behaves more or less the same way. You have a yellow, a sort of red, and blue area. So exo, meso, and endocuticle, maybe. And then if you look at, say, Uricantha calcarata, that's the stick insect, it's blue everywhere, and then a couple of layers are red here or there. And then with the praying mantis, it's all over the place. It's pink and purple. Like, this makes no sense whatsoever. You can't compare these things. Now, if you try to apply the structural definition that is currently used can also run into some problems. I had to invent an entirely new nomenclature and system for doing this, but more or less what I went with was, well, I'll show you in just a minute if I can find it. Find the, oh, no, the stained one isn't there. I left it to the end. Okay, you'll just have to find out. What I ended up doing was delineating it using an exocuticle and an endocuticle, regardless of the number of layers. And the way that I did that is based on staining and also structure. You have to use a confluence of factors. So if you just try to apply the staining, you get nonsense things. You'll have, and the reason for this is that some parts of the cuticle, even if it's clearly the same layer, will stain differently. So for instance, this is a beetle cuticle. This is a study done in Germany on I think it was a jewel beetle, if I'm not mistaken. You can see that the exocuticle mostly stains yellow, and then there's an area that kind of stains blue because it's a bit softer or something. And they're saying now this is endocuticle, but that makes no sense because if you look at the fibers in this, they're all the same. There's no notion of homology here. This is just based on, this is just up, the color is blue, it's it's endocuticle. That's it. So that, yeah, it doesn't really work. And the reason we know that it doesn't work is because there have been other studies in beetles that show that these extra thick, chunky layers arranged in the bottom parts of all of these cross sections in different species, those are all structurally homologous. They are all endocuticle because they have characteristics that most likely came from a common ancestor. In other words, they have this layered structure and they have these macrofibers embedded in them, right? So we have a much more useful notion of why these should be homologous, and the terminology flows from the homology rather than just basing it on color, which is notoriously fickle. And so this is Van de Kamp's, notion of what different parts of the cuticle are homologous or not. We can see this in Curculios. This is with the SEM. You can see that there's an exocuticle that is, when it's fractured, it appears to be rather featureless. And then the endocuticle is full of all these large bundles of fibers facing in all different directions. So you can easily delimit that. And so as a result, like I said, nowadays what most people do is they say, well, the helicoidal zone is exocuticle, and with the endocuticle, it's that layered stuff. That doesn't work in phasmids, and it doesn't work in most Orthoptera, as I found out. So even that definition fails. So if we look at this particular insect here, this was a study done in 2018 by some of my German colleagues, and let's focus on figure A, the upper right subfigure. What you see is that they found the first set of helicoidal layers on the outside, and they said, okay, good, that's exocuticle. And now we have a large layer where all the fibers are facing in the same direction. And we can infer that because when it's fractured, it looks like there are no layers in there. It's just a mass of material, right? And if we look at the set of layers underneath of that, right, that's a helicoidal ply that's been fractured. So they looked at this and said, okay, well, the outermost layer is helicoidal, or the outermost zone, I should say, domain is helicoidal. So that's exocuticle. Right. So is all of this endocuticle? What do we call this? What material is this? The same? Is this stuff here in the middle? Is that the same material as these other layers down here? What they ended up doing was they called this outermost zone exocuticle. They said this was mesocuticle, and they said, all right, and we're going back to exocuticle. I looked at this and thought that's a bit silly, and I'll show you why in just a moment. What I think is actually happening is this entire zone, all the way where from that first, what they're calling exocuticle, down to the second one, the whole thing is all the same material. The only difference is that in some parts of this region, the fibers are being rotated or not rotated. So there are two different mechanisms. One mechanism lays down a material and changes up the composition of the material. The other mechanism is responsible for the rotation of the fibers. So you have the same material either rotated or not rotated. But otherwise, all of this I would call exocuticle. And this is something that has precedent in literature. There's a guy named A.C. Neville, Anthony Charles Neville, who studied insect cuticle. He was prolific, worked on this probably for about 25 years, published a book, Arthropod Cuticle. And every time I think I have a good idea about cuticle, it's in that guy's book. But everybody treats this guy like he was nuts. I don't know. I don't understand why. They think that people treat his work like, oh yeah, there's so many artifacts in there, but no, his work is actually really top notch. It's just one of those weird things that because it's old work, it was done in the '60s, people kind of treat it like it's not worth anything. Yeah, and engineers are especially bad for this. Anything older than 10 years old is not considered good anymore. Generally speaking. So for those of us who are taxonomists and look at stuff that's like 200 years old, this seems a bit silly, but that's how it is for many domains of science. Anthony Charles Neville noted that the exocuticle of different types of locusts and grasshoppers often had a zone where all the fibers faced in the same direction, so there was none of this rotation happening, and it was sandwiched in the middle of the exocuticles. It was very clearly the same material. And his hypothesis was that this was happening in areas where there was really high stress. So for instance, in like the tibia, for instance, of a locust, the hind tibia, that's under tremendous bending stress when it's kicking itself up off the ground and lifting the body and going through this jump. So to resist bending and fracture of the tibia, which is quite thin, you have these layers of very stiff material that are kind of nestled in there with all of the fibers facing in the same direction, just to give it a little bit of extra stiffness. But otherwise, yes, it's part of the exocuticle. Sometimes it's diagnosed as mesocuticle, depending on which author you're looking at, but I much prefer this guy's homology hypothesis there. And the reason for that is because I did my own work. So on the left-hand side, this is a piece of cuticle cut that comes from mole cricket, Grillotalpa grillotalpa. That's the European mole cricket. You can see a couple of you can see this grid of triangles on there. That's from a mechanical test that I did. It's called nano indentation. Still working on analyzing that data. But the more important thing is that you have a well-defined area that has helicoidal microstructure, and then you have an area down here which has a large number of layers where the fibers are facing all in the same direction within each of these. So you have an exocuticle and an endocuticle that are pretty easy to spot. And the definition of, exocuticle is helicoidal, endocuticle is unidirectional, works quite well for most of the samples until you start to see things like what's on the right-hand side. There are layers in there that are unidirectional. I know they're unidirectional because I looked at them very closely, and all of the fibers with TEM and all of the fibers face in the same direction. You can see them in cross-section, you can stain them. But you can follow these layers and you can see that they are sandwiched within this material. You can see where these zones end, right? So it's an inclusion within the three-dimensional structure. So very clearly it's the same material, it just lacks the rotation that's happening in the adjacent domains. So these are unidirectional zones in the exocuticle, which is not really something that's well defined. So the structural definition, if I said that, okay, only the outermost layer that's helicoidal is exocuticle, all the rest is endocuticle, the structural definition would fail. Why? Because I would say that everything starting at this upper white arrow on down was all endocuticle, when very clearly it's not. At least I would say that this material up here is different than the material down here. They don't belong together. They shouldn't be called the same thing. And the way that I kind of came to that conclusion wasn't just from one set of images. I did a lot of different kinds of imaging, including things like confocal, using transmitted light and standard histology, electron microscopy to look at fine structure, and then Raman spectroscopy to see if there were differences in composition. And shocker, the stuff that I thought was exocuticle is exocuticle, whether or not it was helicoidal or unidirectional. So you have multiple lines of evidence that are used to make this determination, which is what you ought to be doing if you study morphology in the first place. And I had to kind of come up with a system for naming these different layers. And it can be quite complex. If you looked just at this material over on the left-hand side and said, okay, only the outermost helicoidal zone is exocuticle, right? You would be misled into thinking all of this other stuff was endocuticle. So I came up with a notation system to kind of delineate this. It's based on set theory. I won't go into that too much, but I think what you can kind of understand here is that very clearly you can follow where these sorts of layers start and stop. And they can be numbered and they can be labeled and they can be compared one-on-one from organism to organism in this way. So I'm working on trying to get that published relatively soon. Is there a question? Nope. Okay. So this is the sort of modified homology assessment that I made, right, based on these multiple lines of evidence where we have exocuticle and endocuticle, which have different composition, different types and amounts of protein versus chitin. And the difference there really between the subtypes in here is whether or not the fibers are rotated in a particular domain to form this helicoidal ply structure or not. All the fibers face in the same direction to make this kind of featureless looking domain here. And it's a bit complicated because both the exocuticle and the endocuticle can have rotated layers or non-rotated layers. That's more or less it. The problem here is that I've come up with this definition. It works in Orthoptera. It works in beetles. I don't know if this works in any other arthropods because I haven't looked at every other arthropod. And the fact is, nobody would know because there has been no widespread systematic study of arthropod cuticle across most taxa. For instance, what does Mecopteran cuticle look like? Do scorpion flies have this different type of arrangement? Nobody knows. What about trilobites? What about any of the extinct taxa that have been found? When did all this stuff evolve? Nobody knows. Completely in the dark about all of that. And because of that, we don't have any solid homology assessments for what parts of the animals are actually comparable and are derived from a single common ancestor. So the only way to get that kind of information is to have biologists look at this, not just engineers, but entomologists. So people who are specialists in morphology of cuticular structures. And I can tell you right now, after going to the cuticle section in the International Congress of Entomology, there are not very many of us. So we need more people to study this very badly, I think, before we can get clarity on how different types of insects or different types of arthropods have optimized their cuticle for different tasks. So with that, I'd like to acknowledge all of the people who helped me to do not just the work that I showed you here, but a lot of the mechanical work that has yet to be published. So I have a large number of studies in the works, I guess, a large amount of data that has to be now compared a second time because I have a new definition of what parts are to be compared across species. I had a lot of help from folks at the University of Bonn where I was working, but also from the Imperial College of London. They very graciously allowed me to use and abuse their nano indenter. Same thing for the Chawla research group at Purdue. Nick Chawla was on my dissertation committee and also let me use some of his equipment for mechanical testing while I was a postdoc. And then, of course, I have to mention the Hasbrook Insect Collection at Arizona State University that let me use a large number of specimens, especially during my Ph.D. work. So with that, thank you all for your attention. I won't give a shameless plug for the lab that I no longer work for, and I'd be happy to take any questions you have at this time. Thank you.
All right. Andrew, that was great. Thanks a lot. And it just gives incredible perspective on how strong this cuticle material is. So maybe if you would tell us if there are some applications that there could be for this kind of studies. I mean, why would people be able to or use this knowledge, this information to, what sort of things can they do with that that would benefit people or mechanically or some way?
Sure. There are two ways I would do this. For this. So one thing that you could sort of conceive of, here's my difficult to find. Yeah, here we go. I'm going to share my screen for just a second, if you don't mind. Let's do this one. Yeah. This figure right here. So as a biologist, one thing that I noticed was that when I looked at different types of crickets, locusts, or different types of Orthoptera, they all had very similar structure. So if I cracked open the cuticle of the tibia, let's say, of a locust versus a grillotalpid, right, a mole cricket versus some other type of grasshopper or a katydid, they all looked exactly like figure A for the most part. If you looked across taxa, they had the same essential parts. There are features there that are unique to Orthoptera. The feature that's unique, if you look at the bottom part here, that's kind of teal, they have an innermost zone that can grow or shrink, and it's used supposedly, and this was a hypothesis from this Anthony Charles Neville guy, supposedly they use that as a as a storage mechanism for energy, like fat. Like they deposit chitin and protein there, and if they're starving, they'll feed on it. They'll digest it. And so they'll have this inner padding of this helicoidal type of material, just deposited on the very inside of their exoskeleton. That's unique to Orthoptera. Um, Knowing that there are things like that out there, if you give me a piece of cuticle and I see that, I know what organism that came from. I don't have to have the whole organism. I just need a little chunk of cuticle and I can tell you what it came from. I don't even have to do any genetic testing. I can say immediately this was Orthoptera, right? That seems to me like it could be pretty useful for people who are doing identification work, right? Um, Same thing with figure B. So this came from a stick insect. Um, Stick insects are the only organism I've seen that have multiple layers that are unidirectional inside of the exocuticle. All other taxa, they have a single unidirectional layer. So if you see more than one unidirectional layer inside the exocuticle, I would, as of right now, this is the only group of things I've seen that has that is stick insects. So there are cuticle characteristics, if you look closely enough, that are unique to these different taxa. It's like a fingerprint, right? Um, so I would say one use for biologists is that you can do IDs with this stuff potentially. Um, but from the other side, on the mechanical side, these are what are called asymmetric laminates, um, meaning they're not balanced in any way. So there's like one layer that's really stiff, or one zone that's really stiff, and then one zone below that that's more flexible. So if I start bending that, it's going to warp out of plane, right? So it's not just if I try to bend that down, it's going to warp out of plane in the other direction. Um, it's very difficult to predict how those types of laminates will behave, but insects use them all the time. Um, there have been a couple of instances where engineers have tried to take advantage of out of plane effects with asymmetric laminates. There was a case where NASA engineers designed, in conjunction with the Air Force, a plane that had carbon fiber in the wings, and as it flew, as the speed and the load on the wings increased, it would start to bend, and the wings would change shape because they were asymmetric laminates. They would change shape so that at higher speeds it would become more aerodynamically efficient. That seems to me like it's pretty useful, and that's a general feature of asymmetric laminates, and it's not something that's used very often because it's very difficult to work with these types of laminates. I think that if we can understand what rules are being used to govern the deposition of these different types of layers, like why in one body part does it look this way versus in a different body part it looks another way, right? If we can understand the rules that are governing this, we could try to apply that to man-made structures that are made from composites. So that's the full answer there. If that makes sense.
That's a lot. I mean, composites used for quite a lot of purposes. So there's that leaves a pretty long list of things that could be done in terms of constructing laminates and building different types of composite materials. So thank you. Anybody else have some questions? Anything they want to add or ask or mention? You can unmute yourself and ask a question.
I had a
Uh,
question.
yeah,
Referring back,
Marco, go
what
ahead.
did, what did you say about carbon fiber again? I just want to make sure I got it right. Um,
Oh, that it's a fibrous material.
yeah, at first I thought you said that like the cuticle inspired it, like how it the layers and stuff.
Oh, well, I wouldn't say that cuticle inspires the way that we use carbon fiber, but there's an interesting parallel there in the way that the insects deploy um, laminate materials and the way that engineers use them, right? There's a clear parallel. So you have very stiff materials that are, yeah, arranged into layers and then used to make structures, right? So we do this for various different purposes to make, boat parts, car parts, airplane parts. Whereas insects are, their whole bodies are built like that, just with these different layered structures. Yeah. That makes sense.
Oh, okay. Okay. Yeah, that makes more sense. So we just use them in similar ways. Is that
Exactly. If we could understand how the insects are using them better, then maybe we could apply that to the things that we make, you know?
probably. And I do agree with your entire sentiment of this presentation. Definitely think we need to get more biological understanding of this.
You could always jump in. You could always jump in. Just saying.
Exactly. Right. All right. Anybody else have anything they want to ask? Um, I might ask something.
Okay.
So you used quite a lot of electron microscopy to produce the images and stuff, and I hope my students understood that a lot of those pictures showed us of the different structures are all electron micrographs. These are scanning electron, I would take it.
There's SEM, there's scanning electron micrographs and transmission electron micrographs as well.
Okay, you had both, right?
Yeah.
So now, of course, in this next wave of things going on in our world, where where will this kind of work be able to continue if the electron microscope center is no longer operational at USDA?
Good question. What I've been recommending to people for now is we've been having to send researchers to either Johns Hopkins or UMD to use their facilities. So both of those institutions have scanning electron and transmitted electron microscopes.
Okay, so College Park and John Hopkins, yeah.
Yeah.
Well, that
Uh,
makes sense. Yeah.
yeah, there's one device that we had at USDA, which was quite well known as a cryo scanning electron microscope. I don't know of anywhere else that has one of those, but you don't need that for working with Cuticle. There are standard protocols for doing SEM with biological material that work pretty well. So yeah, you can definitely get by with the more than get by, I should say. You can do quite a lot with the facilities at either Hopkins or UMD. That's what I'd recommend.
So the cryo electron device, is that the one that Eric Irby came up with,
More or
design?
less. Yep.
Yeah, I remember Eric. That was a really neat adaptation that he came up with
And
some
very,
time back.
very useful, I will say.
Incredibly useful, and I didn't realize that there's no more of that anywhere else. I would think that maybe some of these big universities would hop on board with something like that and try to get one or make one or do whatever because
They're
it's
they're very neat. They're very neat systems, but they're very temperamental. I don't know if somebody told me that ours is the only one in the Western Hemisphere. I don't know if that's actually true. But yeah, they're not very common. And yeah, part of the reason is that they're great. They're great for looking at biological material. The low temperature makes just the samples they just turn out awesome. But they're very temperamental. Yeah. And the climate has to be controlled just so. Yeah, the gas has to be perfectly dry. There's a lot of requirements for it. It can be very technically complex. Um, you know, versus a normal scanning electron microscope, which is a bit easier to operate, you know, but there's more sample prep involved.
Yeah, that's true. But the interesting thing about the cryo electron is the ability to see
Behavior.
behavior in motion or in action, I should say, frozen in time like that, the behavioral aspects that could be captured um, is something that I mean, it was incredible. I remember hearing about this and seeing some of it that Eric had done. Pretty incredible.
Yeah. Yep. Agreed. The ability to look at things that have been frozen in time, like mites doing stuff on a plant sample,
Yeah.
is yeah. There's really nothing else like it, you know. I've often wondered though if there's a way to prepare samples like that, you know, with freezing and then critical point, you know, I've wondered if it'd be possible to freeze something and then freeze dry it so that it doesn't have to be kept frozen. But yeah, things become when they, you know, when you freeze dry them. So maybe that's not a good idea. But
That's why we need more people to jump on board.
yep, yep.
So anyway, does anyone else have any questions that they'd like to ask right now?
Yes, even though they like suspended actions at USDA, would somebody like Department of Defense be carrying on like the same type of investigations, but like you wouldn't know about that things because would be used differently? Like,
Oh, well, yes and no. So I don't know that DoD has a composites lab actively working on insect exoskeletons. However, they do fund research on insect exoskeletons. So the one beetle that I showed you, and I alluded to this at the very beginning. God, what university was it? Was it It was CSU San Diego, maybe? I forget which institution it is. There's a guy down there working on that. He gets $1 million a year from DoD to study that beetle.
Wow.
Yeah, and I'm just thinking, maybe I got in the wrong line of work.
yeah, I would think like something like that when like you said, your first slide of the beetle made me think about ballistics and just like when we watch movies, Marvel movies and things like that,
Yeah,
the
right.
material for the suits and things like that.
The Iron Man suit. Yeah. Yeah, composites are pretty amazing materials. Yeah, if I had to do that again, I probably would have gone to DoD for funding. Because yeah, they do DARPA. I think it's DARPA that's might have gone. He might have gone through DARPA. I think for the composite stuff. Yeah.
It makes perfect sense. Department of Defense would be interested in that kind of thing for materials,
Yeah.
because I can think of applications like that might be of interest to them. So, and that beetle, by the way, I've tried to pin those. God, putting a pin through one of those and other similar beetles in that group
Yep.
are like you can't push the pin through. I mean, it doesn't work. You will bend the pins every time. So you have to almost like drill it, if you will. Use the pin in a drilling motion. Eventually, you'll make a little bit of a tiny hole in it, and then you keep working that. But it takes a long time and patience. Just pushing the pin through will bend the pin every time. They're unbelievably hard.
Yep. Yeah, those are pretty cool animals, I have to say. I wonder about the thing is for every one of those, right? How many organisms are out there that they're not even on they're on nobody's radar, you know? like
Yeah.
some weird little beetle living out in the desert somewhere that nobody's really, oh yeah, this is a hard one to pin too, but nobody really ever bothered to look at it.
There's probably plenty like that.
Yeah. Yeah. There's a lot of stuff out there that's just, yeah, not very well characterized and also very cool.
Indeed.
And then the
Oh,
question to go along with that is it able to be mass produced? Like,
Now that is a good question. Production, right? So if you were going to go for funding from DoD for something like this, being able to demonstrate the ability to produce it, is like that's the next step, I guess. So studying the natural model is one thing, and then actually producing something like that is a whole different ball game. There was a study that was done on it was for bulletproof vests, and they were looking at Kevlar and aramid composites. And the way that they conducted the study was they tried to mimic the fracture resistant properties of oh god, what was the organism that they were going after? I think it might have been mantis shrimps. So they did this helicoidal arrangement of the layers, right, the fiber layers inside of this aramid composite and in a Kevlar composite. And what they found is that it doesn't impart, it doesn't universally, that structure, that helicoidal arrangement doesn't always make the composite stronger when it's hit by a bullet. It only makes that composite stronger if the fiber is extremely brittle. So if the fibers are very stiff and brittle, having that arrangement definitely is optimal. But if the fibers are very tough, then actually it's better to have a 90-degree cross ply. So, it's a, kind of an interesting thing to consider that, in this case, with insects, the fiber is extremely brittle. So of course they have this structure in there, and it also gives us an insight as to why the endocuticle doesn't have that same structure, because it's much tougher. The fibers are less brittle, so they tend to be bundled in these large bundles and have cross ply arrangements. So, yeah,
very good.
something to think about.
All right. Well, I have taken the attendance for the students. So anybody of the students, if they need to log off, they're welcome to. If there aren't any other questions, we can let our speaker off the hook, right, so to speak. And again, thank you very much, Andrew. We appreciate you. Everybody put up a clap sign on your window there. And thank you. I'll clap. You can hear me. Appreciate that so much. Thank you for speaking for us. We really appreciate you coming out tonight and doing this for us. So,
no, thanks for having me. Much appreciated.
Absolutely. I will hope I'll continue seeing you through the Entomological Society of Washington. I'm assuming you're going to keep your role there for the time being.
For as long as I can, yes.
Yeah. Okay. So hopefully I'll see you down there one of these days too.
All right.
All right. Okay. Thanks again. Right. I will ask the MES members, the few of us that are here, maybe to hang